I am Dr.S.Jayakumari, Professor, Department of Pharmacognosy, School of Pharmaceutical Sciences, Wales Institute of Science, Technology and Advanced Studies. Today I am going to talk about the hot continuous extraction by SOXHLET apparatus. Extraction is the process of separation of the soluble component from insoluble plant material. The hot continuous extraction is a type of extraction. Here the method used is a continuous hot method. The apparatus used is a SOXHLET. It is an assembled apparatus. It contains three components. The number one component is a round bottom flask. Number two is the extractor where the main extraction is taking place. Third one is a condenser provided with the inlet and outlet. So here the principle involved is here a complete percolation of the plant material by with the solvent, and continuously the solvent was evaporated and distilled and condensed, again it was rotated. So it is a continuous process, that is why it is called hot continuous extraction. In the RB, the solvent which is going to be used for extraction was taken in the solvent. The plant material which is going to be extracted is taken in the extractor in the small pouch which is made up of either muslin cloth or Wattman filter paper, that is called thimble. Inside the thimble, the plant material was coarsely powdered and it was packed, then it was inserted into the extractor. The extractor usually it is a cylindrical in shape. It is provided both outlet and inlet. In the basal part it is a long tube. Here there are two side tubes, two tubes are attached. One is a side tube, the other one is a small siphon tube. Then above the extractor the condenser was placed. The condenser, it is a bulb type of condenser. It provided with the outlet, the excess water will be removed from the condenser. It is an inlet. Here the water was circulated around the bulb of the condenser. It was cooled whatever the solvent, hot vapors coming solvent vapor in the bulb of the condenser. So all the apparatus was set up in that way. First RB, then extractor, then condenser provided with outlet and inlet pipes. The material was packed uniformly in the extractor. Then it is a heating source, it is called mandle. The temperature can be controlled as per the temperature of the boiling point of the solvent. So it was heated, the solvent slowly evaporated into a vapor. The solvent vapor slowly passes through the side tube, it entered into the condenser. Once it reaches half of the condenser, it was cooled by the circulated water, then it became a liquid, then it slowly dropped in the extractor. In the extractor, the solvent was slowly filled up to the 3/4 of the extractor tube. So it was here only the extraction takes place. Here the solvent is completely immersed with the plant material, so a complete percolation takes place. Once the solvent reaches up to the siphon tube, up to the height of the siphon tube, due to pressure gradient, it was siphoned out through the siphon tube into the RB again. Again it was heated, again evaporated, again circulated through the siphon side tube into the condenser. Where condenser it was cooled, again it can be entered into the extractor where extraction takes place. Again it can be dropped into the extracted solvent, dropped into the RB. The process was continued till complete extraction takes place. Usually the duration of the extraction may be 24 hours, 72 hours or even sometimes 3 weeks, depending upon the nature of the solvent, depending upon the nature of the material. So after complete extraction over, we can determine the complete extraction. The solvent color will be very thick in color and it was very thick liquid. With the help of that coloration and with the help of the plant material, we can determine the solvent process, extraction process was completed. Then after complete extraction, the solvent was removed from the RB, then it can be evaporated under vacuum evaporator by using rotary vacuum evaporator. Thank you to one and all.